me here. On the other hand, I like to apologize because I think this is quite a weird talk concerned to this seminar, but I'm forced to do so. So therefore, I try my very best to give you an introduction into peptidomics, which for me is really a long time. Therefore, I added one word in here, which is history, and some key strategies and applications. And with that, hopefully, does that work? No, this is still. Yeah, no. And with that, we go back to the future somewhere within my talk, and finally, in the end, to the next talk of Andreas Zellner, who is really, I think, giving an academic talk. So my part is now here giving an introduction to that concerning history and what peptidomics is about, because many people do not know, and this is, yeah, somewhat this is a shame, because it's really an interesting field. But first, back to the future. I guess most of you, concerning your age, won't know the film anymore. It's the films. It's a trilogy, very interesting. And I have a few quotes from Marty McFly and Doc Emmett Brown within my talk. Why that? Because of several reasons. Huey Lewis and the News is one of my favorite bands, and they appear not just with The Power of Love, but also with Back in Time. And Back in Time for me is like getting back to Hanover, where I started studying chemistry in 1985. This was the year of the film. So somehow this fits in together. In 2000, we introduced peptidomics as a new word into the academic field as a company, BioVision. I left behind peptidomics several years later because of reasons I will shortly introduce within my talk, going to, at that time, Hochschule Weihenstephan. And then there are many links to how we get to the future. One is 2011 with a tiny little peptidomics biomarker study. Get back to that later. Tina already introduced that I wrote review, and with that edited these two books about peptidomics methods together with Lloyd Fricker from New York. And now also have a module about peptides and peptidases at the HSCT. And hopefully soon after 2024, I will enjoy blood peptidomics 2.0 at TUM, and I have nothing to do with that. I can just enjoy that. So a bit about the contents. I will give an introduction about history and concepts compared to proteomics, because most of you will know proteomics. We'll give a few peptidomics applications. There are too many to give them all. Talk about peptidomics of body fluids, as we have done that in old history, and some of the biomarker pitfalls which are still around, especially in the academic field. And then I will conclude and hand over to Andy. The history of peptidomics starts with peptide research. At that time, people just were looking on, but they didn't know about peptides. And the first peptide they found in 1901, which is now more than 120 years ago, was secretin, a small peptide that regulates, for example, gastric motility and acid secretion. And you see 27 amino acids. This is just a little piece of three-dimensional structure, and this is the precursor of that, because typically peptide hormones are cut out by peptidases into the active form. So therefore, we are looking for those native peptides that started in our institute, and we try to do our best in the company later on. And not to focus too much on the bigger ones, on the proteins which are secreted in cells. So therefore, we were mostly out of the cell. Many of you maybe work more intracellularly. My whole thinking is extracellular. And what is the most prominent peptide in the field? That's insulin. Everybody knows, I guess, which was discovered some 100 years ago by Benting and Bess, as is shown in this picture, while treating dogs with pancreatic juice. They found out that they could heal somewhat diabetes, and with that they came to the idea that there should be a hormone in there. They could cleave it by proteases, but it took 30 more years to discover its molecular structure, because sequencing just started with insulin in 1953. And soon after, or almost soon, in 10 years later, also secretin could be sequenced. So with that, we could know about the molecular structure of peptides. And Tina already introduced that there is no clear-cut definition in between proteins and peptides. I missed this slide out. It's too long, but for me today, I would say roughly up to 100 amino acids, I could call that peptide. I will give some idea because why this is some borderline. Insulin is 6 kilodalton, so roughly 60 amino acids. If you take the FDA definition, then insulin would be a protein. So it's quite a bit weird, this differentiating between peptides and hormones, because everything is a peptide that is is made from amino acids with a peptide bond. So dipeptide is already a peptide, and it could grow as long as an antibody, but at some time you will say it's a protein. And why is that? This depends on history of proteomics. Proteomics started with 2D gels with isoelectric focusing and SDS page. And because of that, for technical reasons, you had a lower mass limit that was about 10 kilodaltons. And that was our idea to complement 2D gel electrophoresis, which was up to 2000, the standard technology, you might not guess that, for proteomics. Yeah, we started with mass spectrometry in parallel, but nevertheless, it came after the 2D gel at that time. And our idea was, first in our Institute for Peptide Research and then in the company, to see within this picture, in this terra incognita, where the gel doesn't see much spots in there. Most are in between 20 and 200 kilodalton. With that, you could have roughly 2,000 spots. So this was, maybe we could think of this and save that, because this is the same amount we try to have in our peptide maps later on. Yeah, typically 2,000 spots with many major technical hurdles written in a Nature review from 1999. The biggest problem was that a gel is something that is not really robust from its chemical composition. So therefore, it's really difficult to reproduce spots really exactly and to know whether this is then a new protein if it shifts, or whether it's the same protein that just shifted. And mass spectrometry is much better, but however, at that time, nobody cared about peptides. Yeah, and especially native peptides, although we knew about secretin, insulin, and many more. Yeah, many more are given in this slide. And you see here that this is a molecular mass range from 1 to roughly 20 kilodaltons. So somewhere here is the borderline for peptides for me for today. At that time, we were more with 20 for marketing reasons, but I think 10 is a good borderline somewhat. Up to there, you find interesting components like insulin, also chemokines, which are below 10 kilodalton, and very well-known peptide hormones like angiotensin, regulating blood pressure, oxytocin. Maybe some already know that you could have this as a nose spray even. And next to insulin and glucagon, I didn't mention that is in there. A new peptide came up with GLP-1. I will give some information later on about that. So many hormones, neuropeptides, oxytocin, for example, antimicrobials, I haven't shown in here, or cytokines are within this peptide field below 10 kilodalton. So really very interesting. Also for diagnostic reasons, for example, beta amyloid, which is still in discussion within Alzheimer's, even though we do not know it's really its function in there, but it seems to be quite somewhat indicative for Alzheimer's disease. Could be bioactive components, but it could also be more diagnostic or diagnostic components within this peptide field. And as it is regulated for usually within minutes of action, blood pressure or eating stuff is within minutes or at least below 1 hour, half-lives of peptides are usually that low. And so therefore they are indicating biological changes which are not really very fast, but fast. So they are somewhere in between. Therefore, I also like the peptide field. Yeah, and they are if you look for literature, then you find out that they are involved in almost, and I guess maybe in all physiological processes, as well as diseases. So they are also relevant, not just from an academic point of view. The term peptidomics, in fact, got into the world by me, as far as I know that. Genomics was coined in 1986. Proteomics in 1994, and with this knowledge and dealing with peptides, I thought, okay, with the mass spectrometer, we see that many peptides at the same time that we should also call this peptidomics, what we are doing. And that was given as a talk a long time ago in 2000 at an ABRF conference. They are still out, but I don't know whether people know that. ASMS now was at that time already the biggest conference. But this was the conference where you could meet people who were that old like I am today, with lots of experience in experimental technologies and therefore get really lots of insights. Therefore, I visited those rather small conferences with roughly 500, 600, 700 attendees at that time, and the name or the global title of the conference, From Singular to Global Analysis of Biological Systems, that fitted and inspired me to say, okay, just take the word peptidomics as the first word for the title. And as this was published, at least a few years ago, now it has gone. The abstracts were in the web for quite some time, up to 2017. This was the first written example, but nevertheless, many other people also started with peptidomics. We were not exclusive. In 2001, in parallel with the publications of the human genome, several groups of different groups introduced peptidomics with papers, or at least 3 are papers, and the Japanese one is an okay, it's also a paper, but I cannot read because it's really in Japanese. And groups from Netherlands and Belgium, they also introduced the word per Hart, Kleinenberger-Mann-Scholz, really important people in the peptidomics field. A Japanese group around Minamino, and our company also had a paper at that time. We were invited by Matthias Mann at that time to contribute in a supplement to Trends in Biotechnology, where we try to explain the peptidomics of body fluids. I come back to that later. First, some numbers, and to see that really there are only still a few groups out that do peptidomics. We see that this is somewhat really growing field. Since 2000, 2001, the first 4 papers, this is growing to around 400. This year, I just looked it up, this is a bit above 400. Or not this year, 23 is above 400, we're now at 200 and something. So will be about saying it's growing, but compared to, and these are 3,000 up to 2023. If you search literature, then 3,000 articles is not that much in biochemistry. So this is really a niche. And you have to exclude peptidomimetics because that is different. This is chemistry and not peptidomics. But if you compare that to proteomics, then you see there's a factor of in total of about 60 in publications up to now. And if you see that more adjusted, if peptidomics and proteomics started much earlier, then we may compare 2014 with around 10,000 papers, then it's a factor of 25. So most people do proteomics. Only very few weird persons care about peptides and peptidomics. And why is that so? I wonder about that because there are really important pharmaceutical products. Insulin, this is an old table, in 2015 had $7 billion in sales, or yeah, it's dollar in sales in the diabetes field. So it's really, really important pharmaceutical. We have Leuprolide in cancer, so by Abbott, also in the blockbuster range. And at that time already, GLP-1 was a blockbuster. I will say a few words more in a few seconds. In total, at that time, we had 60 components in the pharmaceutical market. So the pharmaceutical industry already knew that peptides are really interesting for them, even though they have problems with that, but with proteins, you also have problems. And the interesting thing is from seeing for or watching the last years in the stock market, two companies grew up as big as you couldn't believe, yeah, but as in other fields in the stock market, Novo Nordisk and Eli Lilly, two rather medium-sized pharma companies, are now the biggest pharma companies because of just one peptide agonist, GLP-1. So they grew up. Novo Nordisk in Europe with more than $500 billion is a really impressive market cap, and Eli Lilly with 650 in the US is even bigger. So these companies are now really driving the stock market in the pharma field. And I hope and guess that maybe peptides will get more attention with that. So it's in principle, it's just one target, GLP-1, or two targets, another peptide, GIP, in the case of Eli Lilly. And so then we are at the technology side. What's the difference between peptidomics and proteomics? It's not just peptide proteomics. In many cases, yes, it's peptide proteomics, but in several parts, not. So if you look at the general schemes, proteomics and peptidomics, it looks almost the same. The difference is that we do not start with an enzyme into the analysis. And that's what I guess most of you will do, is trypsinize everything you see. And with that, you lose important information, namely the cleavage sites. And the cleavage sites are most important to know which exact peptide, GLP-1, is really bioactive, because the precursor is the glucagon precursor. It's much longer and contains several peptide hormones. And you have to know the exact mass of the bioactive component. And this is the most interesting thing, one of the most interesting things in peptidomics, and really important information. So further digestion within the analysis has to be prevented, which is sometimes really very difficult if you come from brain, for example. So this is one of the main differences in between. The other, so we are too small for informative tryptic digestion. I will show a few examples about that. So we do not get into interesting subpeptides. So we do not add information. The other thing is that as we only have one peptide, it's getting more complex, and as this usually has not a basic residue at the end, the MS&S spectra look differently. They are more complex. They are more difficult to interpret. So these are the two main differences that we just had for one peptide, and that we do have to prevent any proteolysis and never think of trypsin in the first moment. And what is nice with peptides is that they could be easily handled by LC. You know that from tryptic peptides, even if they get a bit bigger, up to around 10 kilodaltons, usually with RP, HPLC, you have no problems. But as it's just one, I introduced that it's more complicated in MS&S. And disulfide bridges, which are typically opened up and kept within peptidomics, you could decide whether you do this way or the other. Both make sense depending on the questions. So a few examples for trypsinizing peptides. This is glucagon. In that case, it would lead to some nice tryptic peptides. But if you look at, for example, angiotensin, there's only one arginine in there. So there's no really more information, but you will lose the information of the N-terminus, I guess, because it's just a dipeptide in that case. if you go for GLP-1, for example, there the information at the C-terminus might be lost with trypsinizin because these are also just small, even just one residue, no, two and one residue, yeah, you will find there. Or what is also possible, the C peptide, which is the processing piece of insulin, has no arginine or lysine at all. So there, trypsin will do nothing. And the other thing is, what will you do with things like that? When you have lots of disulfide bridges, do you open them up and do you cleave that? And you get the same problems like in here at the end. So typically in small peptides, arginine and lysine are not that evenly distributed, or at least you do not find, as in proteins, 1 or 2 or 4 or 5 peptides that are nice, and the rest you can forget because you have enough with that. Yeah, we only have 1. And oxytocin is an example with a disulfide bridge and no basic residue, so also no need for trypsin. You have already seen the 2 books, so therefore I do not have to introduce so much. The only thing I have to move, may I move this away to show Lloyd, because I would not have done this project without him. He's really the expert, still active in peptidomics at the Albert Einstein Institute in New York. He's an expert in neuropeptidomics, and we both together try to find authors and to edit these books in 2018, and that's now the new edition in 2024. A few examples there, what is important, how to discover new peptides. This is more general topic. How to quantitate them, there Lloyd is the expert. Then clinical use is introduced within these books, or modification-specific analysis. Then bioinformatics is a topic that has grown a lot in the new edition, and what has also grown a lot is immunopeptidomics as a new field within peptidomics. Although it's that small, we even have subfields that are working quite well. And sample types in there are model systems, like you may know. What is quite interesting, that there are many animals involved. I will have a further slide about that, why these are interesting. And not that many people deal with cells or with human samples. So what is done here with blood is quite unique in the world. And what's even less done is food stuff. Some people work on milk, yeah, or hen egg, so really interesting things. And plants, I guess this is growing, but I only got the same chapter like in the first one about moss from a Russian group. I guess this will grow in the near future. And also here there's a project working on that. So glimpse of the diversity of those chapters. To give some examples, yeah, stabilization of brain tissue and extraction means that you really have to stabilize for proteolysis. This is the group of Per Andrin from Uppsala. The group of Albert Lebedev from Moscow. We now moved with a complete group to China because of political reasons, I guess. It's really the expert for de novo sequencing of natural peptides, which is quite complicated because of getting bigger, and also with disulfide bridges, they have lots of papers. Quantitative, I already introduced Lloyd as the expert, who gives general considerations, but we have also specific chapters about that. What is quite interesting, because it's against all the textbook knowledge, that there's a modification with D amino acids. Most textbooks tell you that there are only L amino acids out there. No, that's not true. They are used as modification, and Jonathan Sweetler this is one of the biggest important peptidomics groups for decades in the US. He wrote a chapter about that, also another one. And neuropeptides in C. elegans, one person will know that chapter quite well, but it's the group of Mr. Temmermann from Uni Leuven. So also brain peptides are interesting, and deep learning assisted analysis of immunopeptidomics. There's this topic again. There's even a group next door here from computational work in the chair of Bernhard Küssler, Matthias Wilhelm, who introduced this. What I find is really interesting because this is also a growing field using artificial intelligence within peptidomics also. And you see that this is really spread over the whole biology. And yeah, there's one of those quotes. Yeah, I guess that this is nothing for you, but maybe your kids will like that. Because I think this is really that interesting that we should follow this with more intensity. For example, if you look for what I call peptide warriors in my module, we have lots of fighting peptides. I don't know how many of you know that 50% of the bee venom is consisting of just one peptide, melatonin, which is, as you know, quite impressive in its bioactivity. And with that, there are lots of animals that are able to sting or bite. So therefore, they inject peptides, which is the typical problem in pharmaceutical applications of peptides, to get these things in. So melatonin is one example. The snake, and they all, some of them lead to pharmaceutical products like ACE inhibitors, angiotensin converting enzyme with the snake. Ziconotide for pain is from this cone shell, or the GLP-1 processing is blocked. The processing by the dipeptidase is blocked by an exendin from this Gila monster. So also doing groundbreaking research in peptide in the peptide field might lead to interesting compounds, and therefore peptidomics within these organisms would be interesting. And just with spiders, we have 42,000 species in the world. So there's enough to do in peptidomics. And now I have to look for the time. I need to speed up to get back to what we have done in the company. So jumping into 2001, this is a comparison by MIDI mass spectrometry, a sample without modification and after modification. And what we wanted to see is that most peaks are at the same place and even with the same intensity. And some, and in this case, a very big peak, disappear and another one appears, which is shifted by mass. And as we just used iodoacetamide, you know, this is on top of cysteine. So it's just the normal cysteine disulfide reduction and then capping with iodoacetamide. And we expect that this will be multiple times of 58 or better 116 for a disulfide bridge. So we know that this peak has 3 disulfide bridges, and that was an example we already knew. This is beta-defensin. This is a peptide warrior from humans fighting against antimicrobials. And with that analysis, where we just want to see that the method works, we already saw another peak that shifted, and the mass difference in between there is an amino acid. So that was a variant of this beta-defensin, which we just identified by setting up the method. And with this sample, it should show that this also works for very complex samples. So with peptidomics, you don't want to see just 10 peaks, but better 50 to 100 in one mass spec. And with that, MIDI is really very good. Yeah, and you could, if you combine this with liquid chromatography, at that time offline, we ended up with those maps. And if you compare these, yeah, then you see differences, and that was the idea. Yeah. Peptidomics using that to have a 2D display and find differences. Yeah, and all these lanes are multispectra. So this is only a part of the view where every multispectrum is just shifted by 90 degrees, and then you see just on top of the peaks to make it look like a gel, because most people understood that at that time. So coming from a gel type view, but having all the data already in the computer, we could go back to this, or we needed to go back to the samples, because at that time, electrospray LC-MS combination was a pain in the, and therefore we needed to start with MALDI. That was my decision. And I guess at that time it was all right. Today I would change that. Got back to the samples, went into the electrospray mass spec, went for the typical MSMS analysis with a bit more complex spectra. And with database searching, usually you could annotate your peaks within this gel-like view. at best those that were different. And how complex could that be? Is this example which we published? This is finally we found out that this is really written. But anyway, we had an 88% automated sequence coverage with at that time, another quote, around 12,000 in resolution. Yeah, Doc Bennett would say, "Great Scott." You would say, "What? Great? 12,000 in resolution," but this is sufficient to do analysis like this with 2 charge states. With manual confirmation, you could even get to 92%. You see most of the sequence, and it's quite clear it could be no other peptide. So no need to go for trypsination up to 10 kilodalton. And now I'll hurry up. Maybe I skip a few things, but this is important. Yeah, with that in mind, we had a project dealing with these OB mice. Don't know whether people know this in here. It's a diabetes model. You find insulin in there. You see glucagon fragment, another peptide, hormone amylin, even a company has that name. And the interesting thing was because this is a diabetes model, we thought that insulin is changed, and now this is a color-coded view. The doubly charged insulin signal is not really going up. But what we see is that the proinsulin signal goes up. So the pancreas produces much more proinsulin, what are these mice lacking is a CPE, is a carboxypeptidase, so they could not process this is the intact insulin, but the arginines that are sitting at the end of the molecule are still there, and these are biologically inactive forms. So therefore, another carboxypeptidase D with lots of proinsulin leads to the same concentration of insulin. But you have these misprocessed forms, which usually do not appear in that concentrations in normal mice. So these are in red, and in the LC it's also quite clear they're a bit more polar, so they elute a bit earlier. Higher in mass, just an arginine in between, and eluting a bit earlier. So with that, we see the normal textbook knowledge, and somewhat at that time, I could say, okay, peptidomics works. In principle, it works. And then now I skip because of time. I knew that this will work in, forget about the history of doing peptides. What we did then in the company is downscaling. I upscaled at the beginning in the early history from 2 liters to 10,000 liters. We then downscaled from 2 liters to 2 milliliters, which is nice with HPLC. And with that, we could have maps of blood plasma. What we try to go for the pharma companies. This is the group there at that time, with me a bit younger. And I skip this because I think Andy will tell that about sample numbers. I guess that I have need too much time. So I skip all this. Yeah, you could do statistics about that if you have everything in the computer. And what our biggest project was this Alzheimer project. So we moved to CSF because of our customer. In that case, Roche. I guess I can say that today. And finally, we came up with lots of white spots, which are similar for patients with Alzheimer's and others. We found things that increased and also that decreased, the normal thing you do in proteomics, but now for intact peptides, this is going up to here's 10 kilodalton around there, and here's 1 kilodalton starting. So much bigger than triplet peptides, but the final thing is this should not be validated. So therefore, it got into a nice publication, not really highly ranked. And this is really painful for me to see it again, because that was the starting of because this project didn't work any further, no validation. New market broke down. These 2 towers broke down. Celera burned $1 billion. And with that, nobody wanted to invest in genomics, proteomics, or even peptidomics. That was the decision then to go to Weihenstephan and to change into teaching. because our investors got mad at that time. So now I skip the rest because I guess that you will tell that how to do good biomarker stories. Conclusions, yeah, what changed a lot in 20 years is we now have MS resolution that is incredible, and also speed and automation, also in capillary LC, an online capillary LC, which I would not have guessed at that time, where we use 1 millimeter columns, which worked, but not really that well in combination with MS. We now have complete and unified genomic databases. We have more issues in data analysis, but what hasn't changed is that peptidomics still is a research need. I don't know why exactly, but it is with a high expertise. Sample numbers in biomarker studies are still usually too low, but there's still a high demand for biomarkers. And with that, I have to thank people like Professor Forsman, Lloyd Fricker, my partner Peter Schulz-Knappe, Rüdiger Hess, who's still there in Hanover leading the company PX Biovision, Manfred Reider, whom I learned all the mass spec stuff, and my diploma and PhD students, Michael Jürgens at that time, who did lots of method work. With that, I hand over to Marty McFly, or better to Andy McFly. I should tell this